Okay, welcome back. We are talking about Chapter 12 today, and Chapter 12 is all about genetics, and we are really just going to focus on the very first part of Chapter 12, which is about Mendelian genetics. And a lot of the rest of the chapter is kind of focused on exceptions to Mendelian genetics, but we're just going to focus in on that one aspect here. And so Mendelian genetics is so named because it was originated from an Austrian monk in the 1800s named Gregor Mendel. Um, and you've probably heard of this guy before. So he was an interesting character. He actually really wanted to be a science teacher. He was really interested in science, and as a monk, that was not unusual. A lot of monks would act as teachers in local schools. But initially, when he tried to become a teacher, he just didn't have enough formal science background, enough training, and he failed the test that you had to pass to basically get certified as a teacher. So the monastery actually sent him off to university, where he got a really, really good scientific training. And then when he came back, he arguably almost knew too much, and apparently got into an argument with the person giving the exam the second time he took it, and ended up failing it again. So he never did become a science teacher, but he kind of resigned himself to trying to do some of these breeding experiments that he was interested in the monastery in the garden. And so he was working with just common garden pea plants, doing specific crosses there. And he did this over quite a few years, 7 years. And as you can see, he was pretty prolific. So he's estimated to have crossed over 24,000 pea plants. Um, and what he was really trying to test was some of these basic assumptions at that time, mid 1800s, basic assumptions of how inheritance worked. And so one was that each parent contributed equally to the next generation, and that idea was supported by his experiments. The second kind of long held belief was that these hereditary determinants, which now we would call genes, that idea wasn't around back then, but these determinants were kind of somehow blending together in the offspring. And you could see why that idea would exist, because children often kind of look like a blend of their parents. Um, but that idea of blending was definitely not supported by his experiments, and we'll talk about what he did here in just a second. So he did all these crosses with these pea plants, and he really uncovered just some of the key basic laws of how inheritance works, how basic genetics works. So this was huge. I think you can't overstate how important this has been to our understanding of biology and genetics, and even to evolution. So he published this in 1866, but really nobody paid any attention to it at that time. So he had a couple of things working against him. This was right after Darwin had published his big treatise on evolution, so everybody was focused on that. Mendel was really unknown in the scientific community, and he published his article in kind of an obscure sort of regional journal that not a lot of people looked at. He was doing something really different in these crosses, and his article that he wrote was sort of mathy, filled with some equations, and it just didn't look like other common sort of biology articles of that time. So really nobody paid any attention to it, and it took another 40 years really before it was kind of rediscovered. But what he did, what he found, what he laid out, really meshes perfectly with what we now know about meiosis. We were just talking about that, the segregation of chromosomes and genes into sperm and eggs, and he knew nothing about that process, right? Meiosis hadn't been described yet. So it's really kind of fascinating to see how well what he discovered fits in with what we now know about how the actual biology works. And I'll try to tie those things together here in this lecture. Um, so as I mentioned, Mendel was working with just common garden pea. Turned out this was a pretty good choice, probably subconsciously, but ended up being good for his experiments. So a couple of key characteristics of pea plants and his choices that ended up helping him out. So one is that pea plants, as a lot of plants can actually self-fertilize. So the flowers have male and female parts, um, meaning they could, reproduce with themselves, right? So this ended up being important because what that means when you have plants like that is they tend to, more often than not, self-fertilize, and that means they become very, very genetically pure or inbred, right? Pure would be the nice way of saying it. But these plants are very inbred, very genetically uniform. So he had these starting plants where there was just not a lot of diversity, right? So he had this solid, genetically uniform base to start from. Um, and then he also made a good decision in that he decided he was just going to focus on traits that varied in what the book here calls an either-or manner. So, if you're thinking about traits on a pea plant, there might be some where you could have a really wide range of variation in that one trait. But he basically threw those out and he said, okay, I'm just going to look at the flower color because whenever I look at pea plants, they always either have white flowers or purple flowers. There's not like 10 different flower colors. And I'm just going to look at the shape of the pea pod. It's always either smooth or it's wrinkled. So he just picked things like that where there was basically just two different characteristics for that particular trait. And I'll show you kind of the list of what he looked at here in a minute. But just a few terms to clarify that your book uses. So they'll talk about character, which is the actual physical feature. So for example, flower color would be a character. And then that term trait is really referring to the specific form of that character. So a purple flower versus a white flower, those would be two different traits of that one character. And now here we're focused on heritable traits, just meaning these are traits that can be passed from generation to generation. They're not traits determined by the environment, right? We're talking genetics here. So, and I was just talking about his starting plants being very inbred. Geneticists refer to that as being true breeding. So these were very well-defined true breeding traits where, if you had a pea plant that had a purple flower, that that line of pea plants had been purple flowered for many, many, many years, right? That you could go back lots of generations and those plants just always had purple flowers. So that was what he was starting with, was those quote-unquote true breeding strains. And he really looked at 7 different traits. You don't have to memorize these, but I'll show you the list in a minute. And for all these 7 different traits, he kind of followed the same experimental outline, which was he found some of those true breeding parents with those 2 different traits for the one character. So, for example, he would get, if he's focusing on the flower color character to start with, right? He had one true breeding purple plant and one true breeding white plant. And again, that true breeding means if you're to let that purple plant self-fertilize, it's always going to produce purple offspring. If you let the white pea plant self-fertilize, it's always going to produce white offspring. So geneticists are calling those his parental plants, that parental generation. And what he did, instead of letting them self-fertilize, is he would intentionally cross-pollinate, so breed them together, crossbreed. And to do that, he would simply have to take pollen, which is basically the sperm from one plant, and then dust that onto the anther, the female part of another plant. And he would also have to kind of go in and snip off the female part from the plant that he didn't want to allow to self-fertilize. So he's basically taking sperm from the purple plant here and using it to manually fertilize the white plant. And then he would, that fertilized flower is going to turn into a pea pod with the pea seeds inside. He would grow those up, and that would be his F1 or first generation. So parental generation gives rise to the F1 generation, and then he would just allow that F1 generation to self-fertilize. So this plant would just breed with itself, and then he would go one more generation, right, to the F2. So he's basically got these essentially 3 generations of plants in his experiments. And as he's going through these generations, he's just kind of keeping track of, how many plants are purple or white, have the particular traits in question. Okay. And what really jumped out at him is, no matter which trait he was looking at, which character, so here's the 7, right? He was looking at flower colors, I already mentioned, and seed color, the shape of the seeds, color and shape of the pea pods, where the flowers were located on the plants, were they axial or terminal, and then were they basically tall or short plants. Okay, so we had these 7 different characters. And for each of these different characters, he kind of went through that same experimental outline I was just talking about. But what caught his attention is, every time he did one of these parental crosses, so purple with white, for example, he would see one of the traits would disappear. Let me just go back here. In this particular instance, he would see the white disappear, right? So that F1 generation, every plant that he got every time in the F1 always had purple flowers. So it looked like this parent instead of this one. Okay. And then when he went to the F2 generation, so let these self-fertilize, all of a sudden he would see some white flowered pea plants come back in that F2 second generation. And interestingly, in that F2, he was always seeing that white, which we're going to call the recessive phenotype, come back in this very specific ratio. So again, cross purple with white, right? All the F1 plants are purple, so we would say purple is dominant over white. It dominates. We go to the F2 generation, and all of a sudden we get some white back, but we always have about 3 times as many purple, 3 times as many of the dominant as the recessive, the white. So this approximately 3-to-1 purple to white, dominant to recessive ratio. And he was getting something close to that, right, for every character he looked at, right? He looks at seed color, same thing. Crosses them, one disappears in the F1. They both come back in the F2, but in this 3-to-1 ratio. So that jumped out, right? There was some pattern there. And that really suggested that, there was not a blending, right? There was some actual physical particle that was being passed from one generation to the next, and that particle that determined flower color, for example, it could be hidden, it could be masked, but it was still physically there. It was still being passed from generation to generation. And of course, now we know those particles are the DNA, are the genes, right? There's a gene that determines the flower color. He didn't have that terminology. So these crosses that we're looking at here would be considered monohybrid crosses by geneticists. Mono meaning one, just because we're just looking at one particular trait, one particular gene, right? Now we would say at a time. And we always get that 3-to-1 ratio. Okay. And he tried what are called reciprocal crosses, meaning, he would take the pollen from the purple parent plant and dust it onto the white parent, but he would do it the other way as well. He would take pollen from the white parent and dust it onto the purple. And he found it didn't really matter which parent contributed the pollen; he would get the same results every time. Um, so that finding did support that original idea that, yeah, both parents are contributing equally to the next generation. But as I said, that blending theory really went out the window here, because if they were blending, if you cross, your purple flowered plant with your white flowered plant, you'd expect all the offspring to maybe be like light purple, lavender or something like that, something sort of in between the two parents. Um, And then also, as I mentioned, right, the only real explanation for what he was observing is there has to be some physical particle of inheritance that's getting passed from parent to child, which, as I said, of course, is the DNA, is the genes. So essentially, he was seeing this phenomenon of genes without knowing about DNA or about genes. So what he realized was each parent is contributing one copy of one particle, right, one gene to the next generation. And when you've got those two copies together, one can mask, one can hide, one can dominate over the other, but they're both still there. So in modern genetics now, when we talk about Mendel's work and we talk about these kind of crosses, um, geneticists usually just assign a letter to represent each particular gene that you're talking about, and the letter assigned is essentially random. So, for example, we could use the letter R um, to represent the particular seed shape determining gene. And because we have these dominant and recessive versions of that gene, we need a term to refer to those different versions of the gene, and the word that gets used is alleles. We would say there's two different alleles of the gene. There's the version of the R gene that gives you a smooth P, and then there's another version of the R gene that gives you a wrinkled P. And what does that mean? Well, if we think about what we know about DNA and genes, how do you get different versions of a gene? Well, they're going to have slightly different DNA sequences, right? So those different alleles are just versions of the same gene that have slightly different sequence. Uh, and again, one of those alleles is typically dominant over the other. So when we're using this letter nomenclature, um, once you know which allele is dominant, you represent that by a capital letter R in this case, and the recessive would be a lowercase. And a couple of other important terms to throw out here: homozygous versus heterozygous. So as I mentioned, every organism is going to have two copies of every gene, right? We could take it back to meiosis, they would be diploid, right? They've got two copies of every chromosome. So if your two copies are the same, either both dominant or both recessive, big R, little r, we'd say that individual is homozygous, right? Homo meaning the same. Whereas if they have one dominant copy of the gene and one recessive, we say they're heterozygous. And then we can also talk about physical appearance versus the genetic makeup. And the words we use to distinguish these two things are phenotype and genotype. So when we talk about phenotype in genetics, that is, what does the organism look like, right? Does it have purple flowers or white flowers? Those would be its phenotype. Smooth, round seeds or wrinkled seeds. Whereas genotype is actually referring to, okay, what alleles does it have? Is it homozygous? Is it heterozygous? Right? Big R, little r, using letter S in this case. That would be its genotype. So phenotype is determined by genotype, right? We've already sort of talked about this idea. Your genes, right, the alleles that you have are going to determine your physical appearance. So going back to the seed shape example, there's actually two different genotypes that will give you round seeds. Basically, as long as you have one dominant version of that seed-shaped gene, one big R in your genotype, you're going to have that smooth spherical seed phenotype. So there's a couple of laws of genetics that came out of Mendel's work. The first being what's called the law of segregation, and this just simply says what we've already talked about when we learned about meiosis, is that those two copies of a gene that are present in a diploid individual, right? The two copies of the R gene, for example, in this round-seeded pea plant. Those two copies are going to segregate from each other when this individual produces gametes, goes through meiosis and makes sperm or eggs, right? And I'll show you in a minute, but right, that just goes back to the homologous chromosomes separating from each other in meiosis. So it's essentially just saying each gamete produced is just going to get one copy of that R gene. So if we think about the round-seeded big R homozygous dominant parent, right? Basically, each one of its gametes are going to get one copy of that big R dominant gene. And then the recessive little r wrinkle-seeded P parent, all of its gametes are going to get one copy of the little r. Okay. And we can take what Mendel did and put it into the form of sort of more modern form of Punnett squares, which we use to talk about genetic crosses now. And Punnett squares are basically just a nice, essentially mathematical prediction of what the outcome of a particular cross is going to be. And so to set up a monohybrid Punnett square like you see here, you're basically just taking what the gametes from one parent would be, putting those on one side of the square, and what the gametes from the other parent would be, putting those on the other side of the square. And so we don't really need a Punnett square for the F1 generation because the parents were so inbred, right? We basically just know, yep, from this parent, we're only going to get gametes that have a big R. From this parent, we're only going to get gametes that have a recessive R. And we bring one of each together in that F1 generation. And so all of the individuals in our F1 generation are going to have one big R and one little r. They'll be heterozygous, we would say. And because the big R for the smooth P shape is dominant, right? Every individual in that F1 generation is going to look like this parent. It's going to have that dominant phenotype, the smooth P shape. But where the Punnett square really becomes more useful is then if we start thinking about the F2 generation. So, okay, remember from the F1, we're just allowing these plants now to self-fertilize to go to the F2. So we're basically doing this cross here. We're letting a heterozygote, big R, little r, cross with itself, big R, little r. And so now if we think about what gametes are being produced by each parent, well, from this law of segregation, right, basically half the gametes from this P plant are going to get a copy of the big R allele, and the other half are going to get a little r. Same thing's happening over here, half big R, half little r. So now, if we put these gametes on opposite sides of the Punnett square, right, we've got half big R, half little r on one side coming from mom, half big R, half little r on the other side coming from dad. Mom and dad are basically the same plant in this case, right? Now we start combining in the Punnett square. So the square, we bring our two big R's together, big R, big R. The square, we bring big R, little r. Just by convention, anytime you have a heterozygote, you always write the dominant allele, the big R, before the little r. So you would never write it little r, big R. again, right, bring our big R together with our little r, another heterozygote, and then we bring our two little r's together here. And so here, what this Punnett square is predicting we should get is exactly what Mendel was seeing, right? We would predict we would get 3 times as many plants with the smooth dominant phenotype as the recessive. So I do want you to be comfortable with working through a monohybrid Punnett square like this. So if you were given, a specific cross, could you draw out the Punnett square and make a prediction about what the genotypes and phenotypes of the offspring would be? And I'm going to share another YouTube video up on Canvas that'll kind of walk you through the process of doing that, just to refresh your memory and make sure you're comfortable with it. So just to take this back to meiosis again, remember, right, each one of these pea plants is diploid, has 2 copies of every chromosome. Here's that R gene, right? You've got 1 copy of the R gene on one homologous chromosome, the other copy of the R gene on the other homologous chromosome, 1 dominant, 1 recessive. Remember, the chromosomes were replicated before meiosis, right? So we actually have 2 identical sister chromatids attached to each other. But here's our heterozygote, right? Big R, little r. And then if we follow this through, right, here's that law of segregation. So in meiosis 1, we're going to separate the two paired homologous chromosomes into opposite cells. And then in meiosis 2, we're just going to divide that one more time, right? Split the sister chromatids apart. But what we see is exactly what we were just talking about on the previous slides. So, half the gametes from that heterozygote are going to get a big R. The other half are going to get the little r. So the 2 copies segregate from each other. So pretty amazing that what Mendel discovered really fits the biology exactly. And one thing Mendel did, and this is actually really useful today for breeders and geneticists, is sort of testing this whole finding of his by doing what are called test crosses, where he would take a pea that has the dominant phenotype. So we'll stick with seed shape here, right? So a round, smooth pea. And you might remember there's 2 different genotypes that could give you that smooth pea, right? Could be big R, could be big R, little r. So what he was testing is, could I actually figure out the genotype of this plant just by doing crosses? And the answer is yes. He would do what we now call test crosses, where he would take that plant with the dominant phenotype. We don't know the genotype. We're not sure if it's homozygous or heterozygous. And he would cross it with a homozygous recessive plant, little r. And depending on what the genotype of the dominant parent was, you would get 2 different outcomes. So if this smooth peed parent plant was big R, then when he crosses it to the homozygous recessive, all the pea plants that he gets from that cross are going to be smooth. So they're basically all going to be heterozygous every time. Whereas if that smooth peed parent plant was heterozygous, now he's going to get half of the offspring are going to have the dominant phenotype and half recessive. So basically, if he gets any recessive out of the cross, he knows, okay, this plant had to be heterozygous. So that's useful, like I said, if you're a breeder today, you could do some test crosses and try to figure out the genotype of a particular plant or animal if you don't already know it. So Mendel took this one step further and wanted to see, well, do these same rules hold true if I try to look at 2 characters at the same time? So he would try to follow 2 different characters from that list we already talked about simultaneously. So for example, he would look at seed shape and seed color together. And so now, in terms of genetics, if we want to denote that, because we're talking about two different characters, two different genes, we need two different letters. And again, the letters are basically just assigned at random, but we'll stick with letter R for the seed shape. And now we're going to use the letter Y for seed color. Um, and we know from previous crosses that the yellow seed color is dominant to the green seed color, so we use big Y for yellow, little y for green, and like we already discussed, right, smooth peas are dominant to wrinkled, so we use big R for the smooth peas, little r for the wrinkled. But he basically did the same experimental setup, right? So we had these very inbred, quote unquote, true breeding parents that were completely homozygous, either homozygous dominant or homozygous recessive. Um, Crossed those, looked at the F1 generation, and then allowed those to self-fertilize, grew up the F2, and looked at ratios there. And he was essentially seeing some similar patterns, at least in the F1, in that the F1 always looked like the dominant parent, right? So they were always spherical and yellow. And these are called dihybrid crosses now instead of the monohybrid crosses we were doing before, because we're talking about two genes. But in Punnett square form, here's what the cross might look like. So here's your true breeding dominant parent, right? Big R, big R, big Y, big Y. It's got the two dominant traits, smooth, yellow. And then you've got your true breeding recessive parent plant, green and wrinkled, big R, little r, little r, little y, little y. And now each gamete from one of these parents is going to get one copy of the R gene and one copy of the Y gene, right? So this parent is basically going to contribute a big R and a big Y to the next generation. This parent is going to contribute little r, little y. So what we get is one big R, one little r, one big Y, one little y, right? Basically a double heterozygote, heterozygous for both genes here. And because we've got one dominant R gene, one dominant Y gene, it's still going to look like that smooth yellow parent, right? So all the F1 plants are smooth and yellow. And now this is where things get a little interesting. If we want to make the Punnett square to go to the F2 generation, right, we're letting this plant cross with itself. So big R, big Y, little y, crossed with the same. But if we think about what are the gametes from this parent going to look like, well, each gamete's getting one copy of the R gene and one copy of the Y gene due to that the laws of segregation that we already talked about. But now we've got four different possibilities. So we could get big R with big Y, could get big R with little y, little r with big Y, or little r with little y. So you should get these four different gametes being made from this parent in basically equal proportions. And so what we do with the Punnett square is those four different gametes are going to go on each side of the square. And if you had parents that were genetically different, if you weren't doing a cross here where they were self-fertilizing, you would have to think about, okay, what are the gametes? What do the possible gametes look like from each parent? And then one parent goes on one side, the other parent's gametes go on the other side. But now we've got a bigger Punnett square, right? It's a 4 by 4 instead of a 2 by 2. But the basic idea is the same. So we're bringing our two copies of the R gene together, right? Big R, bring our two copies of the Y gene together, and so on. And so just by pairing them up, we could predict what the genotypes are going to look like, or what the phenotypes are going to look like. And what caught Mendel's attention here was, instead of that 3-to-1 phenotypic ratio that he was seeing in the monohybrid crosses, right, 3 times as many dominant as recessive, here he was getting what we call a 9 to 3 to 3-to-1 ratio. What does that mean? Well, every time he did one of these crosses, he was getting basically 9 times as many plants that had both dominant phenotypes, right? So smooth yellow, look like that parent plant. 9 times as many of those as he was of the uh, plants that look like the recessive parent, so had both recessive phenotypes, wrinkled and green. Um, but then he was also getting, he would get like 3 that had one dominant phenotype but one recessive, right? So they're green, 3 green, which is recessive, but they're smooth, which was the dominant shape. And then likewise, he had another 3 that had the um, dominant color, yellow, but the recessive shape, wrinkled. Okay, so 9 to 3 to 3 to 1. He kept seeing that same pattern over and over again, right? No matter which 2 traits he was following in combination. So 9 to 3 to 3 to 1 phenotypic ratio was characteristic of these dihybrid crosses. And again, I'm going to share another YouTube video that kind of walks you through how to set up and work through these dihybrid crosses. But on the quiz or worksheet for this section, I'm going to give you, definitely going to give you some of these monohybrid or dihybrid crosses, and I want to see that you could work through a Punnett square and make some predictions. Okay, and so just like we just like we discussed, we could take this back with the monohybrid crosses. We could take this back to the dihybrid crosses, um, back to meiosis, and this brings up sort of the second law that Mendel uncovered, which is now called the law of independent assortment. And this is just referring to the fact that when you're following 2 different characters, 2 different genes at the same time, um, assuming those genes are on different chromosomes, they are going to assort independently of each other. So basically what this is saying is, in terms of meiosis, is one homologous pair of chromosomes, right? This pair of homologs that contain the R gene are basically not paying any attention to this other pair of homologous chromosomes that contains the Y gene. Um, and you might remember we talked about, when we were discussing meiosis, the fact that homologous pairs can line up in one of 2 different orientations, right? It could line up like you see on the left here, or they could line up like you see on the right. And depending on how they line up, you'll end up with different combinations of your R and Y alleles. So again, the bottom line is, because of this law of independent assortment, you're going to, in these dihybrid crosses, you're going to get these 4 different possible combinations of gametes, right? Big R, big Y, big R, big Y, little r, little y, big R, little y, little r, big Y, in equal proportions. Okay? And this just goes back to, that's these 4 that we get, right? Basically all possible combinations of one R and one Y in equal frequency. So again, that's explained nicely by what's actually happening in meiosis, independent assortment. Okay, so I'm going to stop there. I will post up a short second part that's going to talk about how you could actually use what's called the multiplication rule to solve some larger than dihybrid problems like trihybrid or tetrahybrid type crosses. And then I'm also going to show you pedigree analysis, which is used to study human genetics.